I want to talk a little bit about factors that impact how well enzymes do their business. So we talk about activity and how well an enzyme performs. So we have this kind of idea that there's a certain number of molecules the enzyme can act on, and there's certain conditions that will actually allow for that to happen at a faster rate. So let's talk a little bit about chemical kinetics. As a refresher, kinetics is the study of reaction rates. And there's a number of different things that can impact reaction rates, but in general, temperature is one that we always point to right away. You increase the temperature, you increase the speed of things, and that's because of collision theory, right? You bash things together, then the chemistry happens, and if you increase the speed, you increase the motion of the particles by increasing the temperature, then that increases how quickly things can occur. So this is the same for enzymes to a point. So if we're looking at a reaction rate versus temperature, then usually there'll be kind of an increase. There's an increase, and then it might peak, and then it's going to decrease again. So there's kind of this sweet spot here, right at this maximum where there's ideal optimal activity. So this would be the maximum amount of activity that we get from that enzyme. And then we could say, I mean, depending on the different enzymes and depending on the different roles and functions of them, then they're going to react at different temperatures. So we could say, well, this is the ideal temperature. For biological organisms, then the body temperature, whatever that might be, for humans, of course, 98.6 degrees Fahrenheit, then having proteins that work maximally in those ranges is very important. So the temperature matters, which is why when our body temperatures get too low or too high, then that starts to slow down normal processes. So if we get too low through something like hypothermia or something, you're out in the cold for too long, and we go too low, then that's going to decrease our enzymes' ability to do the work that they need to do. Or if we get too hot and we have, fever, something as our body is fighting off various things, then that causes other things not to happen as quickly. So there's a small temperature range where these things work optimally, and that maximum is going to be slightly different for different enzymes. But we would assume that most enzymes that are working in humans or in particular organisms are going to maximally work around the body temperature of said organism. Now, another thing that impacts enzyme activity that isn't something we usually talk about with chemical kinetics in general is pH. So there's an optimum pH in the same way that there's an optimum temperature. There's an optimum pH for enzymes as well. And this is going to be different for different enzymes. So for example, enzymes in our stomach might have kind of a peak that looks like this. There's kind of this optimal pH range. So maybe the optimal pH is somewhere between 1 and 2. We might assume that that's an enzyme that works in our stomachs, for example, which is roughly the pH of stomach acid. So it might be something like pepsin, for example, that works there, a digestive enzyme that helps to break down things. There are other enzymes that are going to work in different areas, different pHs, so we might have things around a pH of 7 or 8, so that works under just kind of normal body conditions. So these different pHs are going to be different depending on where in the body these things are located, what they're doing, and then it's why if they're in an environment that isn't optimal, they don't work as well. This is why we need to have buffering systems in our bodies in order to maintain a pH range, because there's only a slight range. These peaks tend to be kind of sharp in some cases, to where there's really a small range where these enzymes can work and function optimally. And if we can't keep the pH in that range, then we're going to have problems. So the bicarbonate, carbon dioxide, carbonic acid kind of cycle that we have in our blood, for example, helps to buffer our system, and that helps to keep the enzymes that work in our blood in a particular range. Okay, so temperature, pH, and then concentration. Concentration is another thing that we talk about with kinetics a lot. If you increase the amount of stuff that you have, then that should increase the rate of the reaction, because again, we're talking about collisions. The more things that we have colliding together, the faster things are going to happen. So concentration matters. We'll talk about the concentration here of the substrate versus the enzyme. So remember that the substrate is the thing that the enzyme is acting on, and the enzyme is the protein that is acting as a catalyst. So if we increase the concentration of the substrate and we keep the enzyme concentration the same, so I have the same number of enzymes, but I have a lot of substrate, then initially that's going to cause the rate to go up, which kind of makes sense, right? Because you increase the concentration, then you increase the rate, right? So it goes up with increasing substrate. But at some point, if I don't have enough enzymes, or if I only have a constant number of enzymes, it's going to start to plateau out. It's going to kind of not peak, but stay at the same rate. And this cap, this kind of asymptote, if we want to think it that way, so this line past which this cannot go is going to be based on how many enzymes. And I'm putting enzyme and substrate here in brackets, which indicates concentration. So this kind of point past which there's no return is going to be based on how much of that enzyme is there. And if that enzyme concentration is static, if it doesn't change, then it'll plateau, and it'll still be higher if I have a lot of substrate, but it's not going to go higher to infinity. I'm going to be limited based on the number of enzymes. You can kind of think about it like limiting reactants from general chemistry. When we did stoichiometry, there are things that impact how much of something we can do, and that's going to be based on the thing that doesn't change. Okay, now with enzyme concentrations, if we have infinity substrate, so if we have substrate in excess, and I'm looking at the enzymes, then this case is like this. So as my concentration of my enzyme increases, the rate increases to infinity, given that I have as much substrate as I can get my hands on, or as much substrate as the enzymes can get their active sites on. So this is going to be a straight line. It's a linear relationship. So we increase the enzyme concentration, we increase the rate of reaction until we run out of substrate, and then that won't matter as much. But this is kind of the difference between the concentrations. They will both initially increase the rate, but one will plateau out and the other will go to infinity given these particular conditions. So enzymes can be impacted by temperature, they can be impacted by pH, they can be impacted by concentration. A lot of these things, at least the concentration and the temperature, are similar to things that you've seen in the past about chemical rates and the things that impact chemical processes. And pH is in there too because of how important those different pieces and parts are of the protein, right? The R groups and the way that the acid-base chemistry of Zwitterions works, the pH really matters for the functionality of those proteins to act as enzymes. Okay, if you have any questions on this, don't hesitate to reach out. Otherwise, I will talk to you again soon.